From Microbe TV, this is a special immune booster recorded during the Society for Leukocyte Biology conference at Michigan State University in October 2024, starring Cindy Leifer and Brianne Barker, featuring Darren Lee from UMass Chan Medical School.
Welcome, Darren.
Thank you. Pleased to be here.
So we're just here to have a conversation with you about sort of your interest in science and your journey through science, and a little bit maybe about what you're most excited about in your most recent research. So can you just start by telling us, when did you first get interested in immunology?
So I guess it goes back to as a graduate student, I was working in a lab doing yeast genetics. And when I graduated, I was interested in a disease or something that was more human related. Mm-hmm. And when I learned about immunology, specifically ocular immunology, I was just fascinated by the ability of the eye, this tiny little organ that can influence the immune response systemically, so throughout the body. And so when I started looking for jobs as a postdoc, I found a position at Scapin's Eye Research Institute, and Andrew Taylor happened to have a position that was opening up, and the rest is history, so to speak. But ironically, up until that point, I was really interested in immunology, but had never taken an actual immunology course. So it was really a crash course in learning about immunology. And from that perspective, it was a really fun experience because I actually got to see, rather than just from the book, it was a really practical experience of, hey, these are T cells, these are Treg cells, and here's what they're actually doing to disease. And subsequently, when I started up my own lab, one of the aspects of the eye I wanted to capitalize on was the eye, when it's functioning normally, is clear, so you could see the immune response as it's happening. Very non-invasively, you just look in the eye, and we subsequently developed some imaging just using the typical microscopes and cameras that other ophthalmologists use, and combining that with fluorescent reporter mice. So we used FOXP3 GFP mice. We could see Tregs flying into the eye as inflammation is happening, and that was really mind-blowing to see, because I'm a very visual person and like to see research happening, and you could actually see these Tregs coming into the eye at the time of inflammation. And the mind-blowing thing was they didn't leave. They just stick around and persist and expand. And so what we think is that these Tregs that are infiltrating into the eye are setting up shop in order to help prevent inflammation from happening subsequently.
Because inflammation in the eye is actually a bad thing, right?
Right.
So can you talk a little bit about not just that it's not, you shouldn't have inflammation there, but what's unique about the eye?
So the eye is, to coin a phrase, is an immune privileged site. Mm-hmm. Peter Medawar came up with that phrase, I think in the 1940s, doing transplantation studies, and found that the eye you can transplant xenografts onto the eye, and they persist indefinitely. So whereas if you take a piece of skin and transplant it, from a different animal, or even MHC mismatched, it will get rejected fairly quickly. Now, the reason for that is that if you think evolutionarily speaking, the eye is really needed for finding food and thinking about our primate ancestors. You really need that depth perception in order to, if you're jumping from tree branch to tree branch and you can't judge that distance, then you're going to fall out of the tree and a predator is going to eat you. So it's it's evolutionary conserved because you really need that response.
So often when I'm talking to undergrads about immunology and talking about immune privileged sites, we kind of talk about it in a very, there's just no immune system kind of way. But I don't think that that's entirely a great description. You've already told us a little bit about inflammation and FOXP3. So how would you describe what's going on with immune privilege in the eye?
So I think more recently the term has been kind of changed to ocular homeostasis, because you have these immune cells, and actually tomorrow I'm going to show some immune cells, not T cells, but innate or antigen-presenting cells that appear to migrate from the eye and come to the spleen in the absence of any inflammation. So it's thought that, or we think that these are migrating to set up the tolerance. So kind of like what happens with the gut, because you have all these this bacteria in the gut, and you don't want a massive inflammatory response. It's thought that there's dendritic cells that are sampling whatever's in the gut in order to provide tolerance. We think this is a similar mechanism with the eye, where you have immune cells surveilling the ocular space in order to help promote and support this tolerogenic ocular microenvironment.
So the regulatory T cells that you mentioned in the eye actually they are involved in setting up that like tolerant environment, or is it kind of a chicken and egg thing and they're like in response to, or do you have some insight on that?
So there's multiple subsets of Treg cells, and non-immunologists like to say or criticize me for saying, why do you study these immune cells? They can't make up their mind. They're either suppressive or inflammatory, and you have so many different subsets. But what I think is happening is that there's a subset of Tregs that come into the eye and help chase out the inflammation, right? So that's one subset. And then another subset is found in the spleen or systemically that helps prevent relapse. And I've looked at blood from uveitis patients, and there's certain pathways that are necessary to make the Tregs that we find in the spleen. And in these uveitis patients, fewer of the patients than non-uveitis patients are able to make these types of Treg cells that, at least we find in mice, are able to suppress disease. So it's likely that a reason that these patients are chronic because they don't have this specific subset of Tregs. And then when we look at uveitis patients that have been quiet for a long time, I haven't accumulated enough samples, but on average, they show a much greater response or ability to make these Treg cells. So we think it's twofold. One is to chase out the inflammation and clear it out, and then two, to help prevent subsequent inflammation, so preventing chronic disease from happening.
And do you think that what you're trying to learn about these different populations of cells, that you'll find something where you can induce them? And do you think if that is a way you could potentially treat people with this disease? Exactly.
So we're always looking for, the easiest thing is just to give a small molecule that will induce these Tregs. But it's unfortunately it's a little bit more complicated than that. We've more recently started taking cell-based approaches where you take out kind of like the CAR T cell field. Mm-hmm. We've partnered with collaborators who can make CAR Tregs, and we're evaluating if those are effective in suppressing disease. But this pathway that I mentioned, there's one part of the pathway is dependent on suppressive neuropeptides that are found constitutively expressed in the eye. So during uveitis, these peptides are reduced, and then as resolution happens or uveitis stops, then they start coming back up. So, one area we could target is trying to upregulate these neuropeptides during inflammation and try to chase it out much more quickly.
But it sounds like I'm hearing you say that there's a lot more interaction with the systemic response than maybe people had originally thought.
Certainly than I thought.
Exactly.
Right. Yeah. Yeah. And so that's, that's the great thing about being a scientist. And, there's plenty of different directions to go and things to explore. But on the flip side, it's bad for the patient because, it's a lot more complicated. So, but the one thing with uveitis, another aspect that makes it so complicated is there's so many different types of uveitis. And so, it's also useful to explore all these different pathways and different interactions because there may be one subset of patients that respond to, or the reason for their disease is because there's defects in one cell type, whereas another set of patients might have defects in another type of cell type.
Are many of these patients, starting out with eye damage or infection, or is it really mostly some kind of other autoimmune insult?
So there's the majority of patients are idiopathic, which means we have no idea what causes it. But there are some that have like HLA-B27 or HLA-B51. So those are associated with other systemic autoimmune diseases. So B57 tends to be associated with arthritis and potentially Crohn's disease or colitis. And what's interesting is I met identical twins, both with the B27, and one of them had uveitis, and his twin brother did not have uveitis, but had arthritis in his back. So there's clearly another component to that that indicates or causes inflammation in different organs, and it's thought it's some sort of inflammatory response or potentially infectious response that maybe the proteins that are being targeted, the self proteins, maybe more are being expressed in the eye, and some are being expressed in bone tissue or the joints. There's also the molecular mimicry model where the patient may have some sort of viral or bacterial or fungal infection, and then it gets cleared, but one of the proteins on the virus or whatever the pathogen is kind of looks like self, so it mimics self proteins, and then you develop a memory response against this pathogen, and then so now you have all these this immune response against that protein, but because it also looks like self, then months or years down the road, you may start developing uveitis or some sort of autoimmune disease. And those are really tricky to study because there's epitope spreading. So it's thought that the initial insult or antigen that was triggering the disease has since, the immune response has since shifted, and so it may be against another protein in the eye. So it's really tough to study. But some labs have identified some bacteria or viruses that, at least in mice, they've been able to identify proteins that do mimic ocular proteins, and to show that, at least in the lab, they've been able to show that this is possible. So
given that there's so many different ways for uveitis to develop, how do you model that? So what is the model that you use? Because you have these powerful tools that you said you can just look in the eye,
right,
in the mouse, just put them in the machine and just look and see
right,
where the immune cells are, and they're migrating to the eye and what they're doing when they're there. But how do you create that model?
So unfortunately, there's really only one mouse autoimmune model, and that's an excellent prompt so that I can plug that we need more models.
I did not plan that.
So, but the way that we induce it is basically it's a vaccine response. So we emulsify a retinal antigen, a peptide, with a very strong adjuvant, and give that to the mice, and then depending on the mouse strain, it's either 7 to 10 days or 3 to 4 weeks. We start seeing inflammation in the back of the eye. So, and then again, depending on the model or the mouse strain, it resolves either within 30 days or 3 months. And that resolution process has really been the focus of my lab. But other labs have looked at different peptides of a specific protein and found different portions that are a little bit more immunogenic. But other labs have taken the full-length protein and found that really extends the time frame of disease that resolution happens. So what's probably happening is they're generating multiple T cell responses against that protein, and it just takes longer to finally get them all to quiet down. So
once the resolution has happened, are those mice more susceptible to a future insult?
So actually, no. So if we re-immunize them, the immune response looks very much like a naive response. And
isn't that interesting?
It's not, or so my postdoc mentors published that it's because there's Tregs. So in mice, so some strains, or this pathway that I talked about earlier that is needed to generate this Tregs, there's some knockout mice that don't develop these Tregs. And what happens in those mice is upon reimmunization, they show a massive memory immune response. So it can't be controlled because there's no Tregs. And then he also showed that if he adds back or transfers in Tregs from wild type mice, then it brings it back down, and they see an attenuated or a naive response. So that's really been the focus of my work has been to understand these Tregs, and I've identified different subsets of Tregs based on immune checkpoint receptors that have different homing requirements in order to suppress disease. So some of them can function fine if they can home to both to either lymph nodes or the eye, and then others require expression or the ability to home to both places. And then back to the human studies, we've shown that in the uveitis patients, they show a decreased ability to induce these Tregs that can home back to the eye or back to ocular tissue.
So when we think about Tregs, we often think about like the natural Tregs and induced Tregs. So, but then there's also subsets of the Tregs that I think you what you're alluding to that you haven't said is TIGIT, right? That molecule. So can you say a little bit about natural versus induced, their role in this, and then how TIGIT plays in that and what TIGIT is?
Sure. So I guess we haven't done the best experiment to show that the Tregs that we get are induced or peripherally generated, but they have all the markers and they kind of behave as if they're peripherally generated. The difference between natural or thymic Tregs and peripherally generated Tregs is that the natural or thymic derived Tregs come from the thymus. They're also called central Tregs. And then the peripherally generated are Tregs that occur during an inflammatory response. When the T cell gets activated, you get this differentiation of all sorts of different Th1s, Th2s, Th17s, depending on the context of whatever cytokines are present. But you also get a subset of Tregs that are T cells that are Tregs. And so these are the peripherally generated or inducible Tregs. So within the inducible Tregs, I've identified PD-1 expressing Tregs and TIGIT expressing Tregs. And, we're just still trying to figure out what functional difference they have, but they do definitely, so the PD-1s can suppress. They seem to be a little bit more potent than the TIGIT-positive Tregs. And we have some new data suggesting that depending on how they're stimulated, so which receptors, the adenosine 2A receptor has been a big focus of my lab. Mm-hmm. It kind of looks like, this is very preliminary, very unpublished, but we've only done one experiment showing that we get more TIGIT-positive Tregs with A2AR stimulation, and then this other death receptor stimulation seems to be more able to induce PD-1 positive Tregs. Rachel Caspi at NEI has shown that depletion of Tregs during uveitis extends the disease. She didn't focus on peripheral or thymic-derived Tregs, but my thought is that the thymic-derived may be involved in kind of helping out the resolution of disease, which is also a big question because, great, you get these Tregs that can prevent relapse, but, how do you get clinically? The ophthalmologist wants to be able to clear out that inflammation. So, what sort of Tregs are necessary, or what mechanisms are necessary for that?
So the heterogeneous Tregs that you're talking about with PD-1 or TIGIT, those are only in the induced Treg subset, or is there also heterogeneity in the thymic Treg subsets?
I believe there are most likely PD-1 and TIGIT thymic-derived Tregs, but don't quote me on that. You kind of get pigeonholed into your niche, and I haven't looked into all the nuances of the thymic-derived Tregs myself. So,
well, I think we could talk about Tregs forever.
Oh, yeah, absolutely.
But I wanted to ask you a couple of things. So one thing was, what kind of advice do you give for trainees? So SLB meeting is really great for trainees, and so what kind of advice do you give?
For all the trainees out there, get involved with SLB.
And I also did not plant that one. Can you tell us how you are involved in SLB and what it has meant to you?
So I think the first meeting I went to with SLB was 2009, and right off the bat, I established some collaborations that were very fruitful and helpful, and then I didn't make it back until was it 2010 or must have been 2011. And at that meeting, my postdoc mentor didn't make it, so I was on my own to kind of feel my way around, and surprisingly, people were so friendly and outgoing and just willing to help out, which I hadn't experienced it at other meetings. So I just kept coming back and meeting great people like Cindy, and networking is great. But I think there's so many different aspects of being successful. You really have to be able to look at what you need. So I think for me, I really like being able to tell a story. So I like digging into, how did you find that molecule? What's the relevance? So that's helped me. So I a lot of times tell people, make sure you can tell a really great story, have a great elevator pitch. But then some of my weaknesses, I think, are probably networking. And so other people are great at networking. So, they probably don't need to concentrate on that. But certainly being able to tell your story in a convincing way and be enthusiastic about it is really, I think, to me, that's what really sets someone apart, because that enthusiasm comes across into your grant. And, at the end of the day, that's, if you can't write a good grant and get funding, then, it becomes really hard to continue to do what we all love. So, having that enthusiasm and finding that niche and that, the gap, and being able to sell it is, a quality that is really helpful as a scientist. So, but I feel like we could talk for hours advice for trainees. So
Yeah.
Yeah, absolutely.
Did you have any other last questions?
I sort of going back to the very beginning, you sort of started with being in graduate school. How did you know that you wanted to be involved in science in the first place?
So that actually goes back to probably first or second grade. I was always kind of interested in planets. So I would go to the library with my mom, and I'd sit down in the reference section and just like open the atlas of all the planets. And then my cousin, who was a year or 2 years older than me, came over one day and had a book that had all these planets in it. And I'm like, oh, what class is that? And she said, science. And I said, ooh, I want to do science. Just because I was interested in the planets. And then from there, it became, I'm interested in how things work. And then, there's اتوms, and then biologically, how do things work? And so it kind of built from there. So I'd say at an early age, there's already an interest in science. And you could thank my mom for taking me to the library and planting me in front of the planet atlases, and my cousin who, unbeknownst to her, planted the bug in me for science. So then I guess from there it was just, what do cells do? And then you take the classes, and you just get more and more interested in all the teachers that you would ask questions. And I remember in high school that the teachers being asked questions, and they would say, oh, that's complicated. And I'm like, oh, is it really that complicated? Let's dig into that some more. And I'm sure they hated that, but they're just like, oh, you'll learn in college when you take biology. So then, that planted the bug. Okay, well, I need to take biology in college. And it just kind of built from there.
Well, we're glad that all of those things happened and that you got the bug so that you could tell us all this fascinating stuff.
Sure. Yeah. Happy to be here. This has been fun.
Yeah. Thank you for joining us.
Sure.
Really
Thanks.
Appreciate
it.
Thank
you for the invitation.
That's a special episode of Immune and Immune Booster. Questions and comments can be sent to immune@microbe.tv. And if you enjoy our work, please support the science shows of Microbe TV at microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today from UMass Chan Medical School, Darren Lee. Cindy Leifer is at Cornell University. Cindy Leifer on Blue Sky. Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious.